I'm Dr. Ganesan, Assistant Professor, Department of Genetic Engineering, SRM Institute of Science and Technology, Katambattu. In this video, I'll be giving you a brief introduction about ubiquitination, a well-characterized post-translational modification. Proteins are functional components and the regulation is critical for maintaining cellular homeostasis. There are two major pathways by which cells degrade proteins: lysosomal pathway, which involves breaking down proteins inside a specialized organelle called lysosome. The ubiquitin proteasome pathway, which is more specific and tightly regulated. Ubiquitin proteasome system ensures that only targeted proteins are broken down, preventing unnecessary destruction. It is controlled by a multi-step enzymatic cascade involving three key enzymes: E1, ubiquitin-activating enzyme; E2, ubiquitin-conjugating enzyme; E3, ubiquitin-ligase. Ubiquitin is 76 amino acid protein that acts like a molecular tag. When a protein is no longer needed or is damaged, ubiquitin gets attached to it, marking it for degradation. This attachment happens specifically at lysine residues of the target protein. Tagging a protein with ubiquitin is carried out by a cascade of three enzymes. E1, ubiquitin-activating enzyme. This enzyme kickstarts the process by activating ubiquitin with the help of ATP binding. E2, ubiquitin-conjugating enzyme. It transports the activated ubiquitin from E1 to E3. E3, ubiquitin-ligase. This enzyme plays the most crucial role because it determines which proteins need to be tagged for degradation. Once a protein is tagged with ubiquitin, it can follow one of the two pathways. De-ubiquitinase pathway. Some proteins can be saved from degradation by an enzyme called de-ubiquitinase, which removes the ubiquitin tag and reverses the process. 26 proteasome pathway. The 26 proteasome consists of the 20S core, which is responsible for breaking down proteins into smaller peptides. The 19S regulatory units, which control the entry and processing of ubiquitinated proteins. E3 ligases are classified into different groups based on the domains they contain. The major types include Hecht, ring, U-box, culling ring ligases. E3 ligases function two different ways: direct transfer mechanism, where ubiquitin is transferred directly from E2 to the target protein. Domain mediated transfer, where ubiquitin is first transferred from E2 to the Hecht domain of E3 before being passed on to the target protein. Importance of ubiquitination. It is a key regulatory mechanism that controls many biological processes, including plant development. One striking example is in rice, where the targeted ubiquitination of large proteins has been shown to enhance important growth rates. It plays an important role in auxin signaling, gibberellin signaling. Ring-type ligases play a major role in regulating immune signaling by controlling the stability of protein. During times of stress or nutrient limitation, ubiquitination enhances the autophagy pathway, helping plants recycle cellular components for survival and growth. What are the disadvantages of ubiquitination? Ubiquitination is a highly regulated process, but when it fails or malfunctions, it can lead to various problems, including premature degradation of essential proteins or accumulation of defective proteins. Ineffective ubiquitination can lead to buildup of damaged or misfolded proteins, which may form toxic protein aggregate. Some proteins act as a tumor suppressor, controlling cell division and apoptosis. If ubiquitination erroneously degrades these proteins, uncontrolled cell growth can occur, leading to tumor formation. Defects in ubiquitination can lead to an imbalance in metabolic processes. Thank you for listening.